The overall goal of this procedure is to deliver various types of biologically meaningful molecules in a sequential and dosage controllable manner with high efficiency using the Vortex-Assisted microfluidic electroporator. This is accomplished by first preparing 4 50 milliliter centrifuge tubes individually containing DPBS, solutions with cells, and biomolecules, and attaching each tube to its respective vial holder connected to the pneumatic flow control system. The second step is to insert the inlet tubings from each respective vial into the microfluidic device with the embedded 15-pin electrodes. Next, the cells are flowed into the device where they are trapped in microscale vortices formed in the electroporation chambers. The final step is to apply short electric pulses to the trapped cells, promptly followed by injecting the solutions containing the biomolecules to be delivered into the cytosol. Ultimately, the cells obtained from this process can be released and collected for downstream analysis.
The main advantage of this technique over existing methods is that the proposed technique is capable of sequentially delivering controlled amount of multiple molecules into a pre-selected identical cell population with high efficiency and viability.
Visual demonstration of this method is critical as the fluid exchange steps are difficult to learn because the timing of the co-flow step at each solution switching step determines stability of the cell trapping.
A metastatic breast cancer cell line, MDA-MB231, will be used in this experiment.